The microscopic organisms we usually blame for destroying our food are the exact same biological engines we rely on to perfect it. In its natural baseline state, raw food is highly unstable. Exposed to the open environment, ingredients like milk, fresh vegetables, or grains will rapidly succumb to natural decay and spoil. The solution to this instability is counterintuitive. Instead of attempting to sterilize the food to protect it, we intentionally introduce massive colonies of specific beneficial microbes into the raw ingredients. The boundary between spoiled milk and a high-value cheese is a highly controlled act of biological engineering. The scientific study of this process is known formally as microbial biotechnology in food systems. This syllabus from a food science curriculum outlines the specific academic mandate. Scientists must identify and master the use of food-associated microorganisms, specifically beneficial microbes and starter cultures. Starter cultures are the primary biological tools used in this field. They are pure, highly concentrated doses of specific microorganisms that allow scientists to predictably control the fermentation process. These cultures are physically injected or folded into raw, vulnerable ingredients to initiate preservation before wild, harmful bacteria have a chance to take hold. We are deliberately deploying an invisible workforce to secure the food from the inside out. To see exactly how this invisible workforce operates, we have to scale down and look closely at the microscopic level. The first primary biological agent we rely on is lactic acid bacteria, or LAB. These bacteria thrive in specific oxygen-depleted environments. When you submerge vegetables in a brine or seal them away from the open air, you are creating the exact conditions LAB need to wake up and start working. The second major agent is yeast. Unlike bacteria, yeast is a single-celled fungus with a totally different cellular structure and a distinctly different way of processing nutrients. Together, these organisms act as a dual engine for food processing, physically dismantling raw ingredients at the cellular level. This diagram illustrates the biological basis of fermentation. It is a metabolic process where these introduced microbes consume the simple sugars naturally present in the raw food. When lactic acid bacteria consume those sugars, their specific metabolic output is lactic acid. Yeast takes a different metabolic pathway. As it digests sugars, it excretes carbon dioxide gas and alcohol. As lactic acid pools in the food, the pH of the entire environment plummets. This rapid drop in pH is the critical defense mechanism. It creates an environment so highly acidic that dangerous, spoilage-causing pathogens simply cannot survive in it. Meanwhile, the carbon dioxide produced by the yeast gets trapped. The gas expands, physically altering the food's matrix. This is exactly the mechanism that causes a heavy loaf of bread dough to rise. Looking back at the academic requirements, Unit 2.2 highlights exactly this chemical interplay. Understanding yeast metabolism and lactic acid bacteria is non-negotiable for food scientists. Fermentation is an active, aggressive chemical transformation that simultaneously secures the food from rotting and restructures its physical form. Returning to the human scale, we can see the results of this chemical process in the foods we eat every day: yogurt, kimchi, sourdough, and wine. A fragile, highly vulnerable raw liquid like milk has been chemically altered into a solid, stable block of cheese that can last for months or years without refrigeration. During the digestion process, the microbes consume simple sugars and synthesize new compounds like B vitamins and organic acids. This makes the existing nutrients more bioavailable, allowing the human body to absorb them more efficiently. Because of these added vitamins and the removal of moisture during processing, the final product is a new food category, engineered to deliver denser nutrition and prolonged stability. Many of these beneficial microbes survive the entire fermentation process. We call these living holdovers, probiotics. When we consume these fermented foods, those living starter cultures transfer directly into our own human digestive systems. These external microbes supplement and reinforce the human digestive microbiome, actively aiding in our own digestion and supporting immune function. The role of microorganisms in food biotechnology directly translates to a measurable impact on human biology. Food biotechnology is the mastery of an ancient biological partnership. By managing these microbes, we are choosing to integrate an external biological system into our own to sustain and improve human health.